And well, it's good to see a lot of seats filled up. Hopefully, I can make your time worthwhile. So, as she introduced, I'm a bioinformatician at Protein Data Bank Europe, and today I'm going to talk about PDB-KB Ligands. These are a set of web pages we have released in 2024, where we aggregate the information of ligands across the PDB archive. And today I'm going to talk about how you can use this data, not just to download the data, but then you can do analysis, make hypothesis just on the web interface. So before begin doing that, like what do we mean by ligands in the PDB? How the representation of ligands are done in PDB? How are the curation done in PDB? It will give you an idea of like how to look at the data. So I thought it good to just give an introduction on that. So I don't think for this audience, I don't need to introduce PDB. We have heard the good, bad, and the ugly part of the PDB. So I'm going to concentrate more on good part of the PDB and how can you make use of the data. So PDB is when structure biologists identify the structures of the protein, nucleic acid, they deposit the structures. So based on the location of the deposition, this gets processed at different units. I'm part of the Protein Data Bank Europe. We are located in EBI. So if you are from Europe or Africa, those get processed at our site. Irrespective of the location, all the processing are standardized. They get deposited into a single archive called the Protein Data Bank. Now, once it gets deposited, it goes through a lot of semi-automatic curation process. It's not completely automatic. There are bio-curators sits process this data. So one of my colleague, Deborah, is with me. She has recently reached a milestone of 10,000 structures curating it. So if you have any doubts about curation, she's the best person, and I'm sure she knows more about curation. So it involves a lot of process, a lot of manual and automatic process. My talk will touch upon some part of like how the curation happens for the ligands, or what we call as chemical components. So if you think about proteins, these are polymers. So nucleic acid, they are also polymers. So if you want to have a well representation of that, you can break them down to small monomers, right? So amino acids or nucleotides, but the ligands are like also other chemical components. So once you break them down, you can have accurate chemical description of that. So what's the use of such a representation is that you can reuse these chemical definitions. You deposit a new structure, then you can reuse this amino acid. But when, for example, there are a lot of structures with ATP, ADP, you can reuse these definitions. And when a new ligand definition comes, you can create this definition. And that happens with the discussion with the depositor. These new definitions get added to a dictionary called Chemical Component Dictionary. Now, this is represented using a file format called mmzip. This is a simple format where you have key value pairs or the table kind of format. So each chemical component has a unique ID, so that helps you to identify them and search them. And important information in this file is like you have the 3D coordinates. So when a chemical component definition comes, you have a structure corresponding to that, the first structure from which the definition was created correspond to the model coordinates. And then you can create the ideal coordinates using a software. So in the curation, it's use OpenEye software for creating these coordinates. Also, it includes the bond type, bond order information in that. So if you want to analyze what kind of a ligand is, what are the atom types, bond order, these informations are encoded in this file. And then it also includes all a different type of chemical description like SMILE, INGI. Now, what happens, it helps you to reuse these definitions, but then it causes one problem is that a lot of ligands in PDB get fragmented using this process. For example, look at this structure. This is teichoplan. So PDB has grown over the 50 years, and then initially it just was for proteins, but recently when you see a lot of structures get deposited with ligands, and you see different type of representations, and some of the ligands are like which are like called the peptide like antibiotics or inhibitors. It has a peptide core and then monosaccharide. So you have definition for each of them, but then you lose the identity of the entire molecule. You can't search in the database. So one good thing about PDB is we make mistakes, but then we correct the mistakes also. It may take some time, but it happens. And so one of the process happens is like they created a new dictionary called biologically interesting molecule reference dictionary, which takes this kind of molecule which were fragmented, then created a complete chemical description of these kind of molecules. And another one is like carbohydrates. So when I talk to people, like even if we are all in the same community, we are all talking the science, but then people look at different ways. For example, carbohydrates, when you talk to glycoscience community, they want a different representation of carbohydrates. Like there are different representation of their standardization. They look at the linkages between the monosaccharides. And so PDB involved those things. As PDB is a fair database, we wanted to make the representation standardized. So there was a huge effort on the carbohydrate remediation process, which helps you to represent these carbohydrates in the standardized format, which followed by the glycoscience community. These two are manual processes. These are done during the PDB curation process. But at PDB, when we analyze, a lot of structures still got fragmented. For example, I'm showing a structure of chromomycin. So you have these all single chemical component definitions, but then they are connected together. So if you talk to cheminformatician or chemist, this is the entire molecule, but then that got fragmented in terms of how data is represented in PDB. But good thing is like all the information you need to make that complete description of the molecule is present in the file. So the one thing you need to do is you need to identify the fields, put them together. So that's what we did. We analyzed the structures in the entire PDB and identify these kind of fragmented molecules and created a complete definition. This is an automatic process. So, and then we called them as covalently linked components. So this kind of, this create a complete description. So that makes it easy for you to search them using their own chemical descriptors. Now, so for the rest of the talk, when I talk about small molecules, I'm talking about the entire set of these molecules, mainly CCDs. We have almost 50,000 of CCDs now, and then PRDs, which are like the biologically interesting antibiotics inhibitors, and CLCs that we automatically generate. Those are fragmented one. Mostly they belong to the carbohydrate. And they are all represented using the MMZip file, but this is not a common format. Like if you talk to a cheminformatician, they analyze, it's not a common format that they use. So, and one of the tools mainly used to analyze these structures nowadays is RDKit. So what we did is we developed a wrapper around two tools. One is GEMI, which is like the sponsored by CCP4 and Global Facing. It's a good tool to parse ZIP files. So you can parse the data in ZIP and then create these small objects which can be processed using RDKit. So what it helps you to do, whatever you can do with RDKit can be done for all the files in MMZip. So we create a representation of those ligands. So you have the metadata of properties descriptors which are present in the ZIP file. And also you can, right now we create different classes to calculate these descriptors using RDKit, but any utility available through RDKit can be done using this tool. Now I'm just, now that you have some idea about what ligand, how they are represented, I'm going to talk a little bit about like how can you ask different questions about ligand. If you have some interesting ligand you want to analyze, how can you use the PDB resource to ask such kind of questions. So if you have your laptop open, like I would suggest you to maybe follow through so this is going to be some snapshots of the website. So maybe if you can also follow if you want to, or otherwise I will show what the utilities available. So just to motivate what kind of questions you can ask, like I'm gonna take a simple example of tetrahydrobiopterin. This is a cofactor molecule with a biological function. For example, it's a cofactor for creating neurotransmitters like dopamine, serotonin, and it's a cofactor for phenyl hydroxylase, where it converts the phenylalanine to tyrosine. These are all just some information, but all that I want you to take away is this is some ligand we are interested to analyze. So if you know the ligand name, you can go to PDB-org/chem. You can put the ligand name. It goes to URL link. So if you know the CCD ID also, you can directly go through this website, PDB-org/chem H4B. That's the CCD ID for the ligand. So it goes to a web page that we call PDB-KB Ligand pages, which has the basic information about the ligand, the 2D topology of the ligand. So it gives you some idea of what the ligand looks like, and it's the basic information like the SMILES, INGI, key informations. And if you want to know the other synonyms information, you want to search in using these names, you can get that. And now, if you want to know what are the different type of substructures present in the ligands, you can navigate to the carousel to see. We highlight some of the pyridine and macroscopol, which are all calculated using aggregate. So that's, we use the tool I mentioned, CCD utils, to calculate them, but then we provide this information through the web server. And if you want to know how the ligand looks in 3D, you can go to the view 3D. It shows both ideal and model coordinates of the ligand. And just like I mentioned, in this case, this is also part of a CLC. Some ligands are like fragmented, but we have web pages for them. So you can navigate to those websites, see how does that look like. And we also shows from which PDB structure this ligand came from. So you can go and make sure that whatever data we are showing is correct in the 3D views also. So I mean, at the point of is at the end of the day, what we want to do is to navigate to the web pages and come up your own hypothesis and see if that's some direction you want to do your search. We also show the physicochemical properties just to give you an idea about how the ligand looks like. For example, in this case, this is a drug-like molecule. The physicochemical properties, you can see if they follow the Lipinski rule of 5, and then just give you an idea of like whether that's something orally active drug or not. And we also add annotations like functional annotations. These are computationally assigned annotations based on the similarity of the drug, similarity of the molecule. So we find similarity against the drug bank database, and also look at where the ligand binds. So if we have some annotation that a similar molecule is binding to a target, which has some pharmacological annotation, we assign them as drug-like molecule. Like a lot of ligands in PDB they are like buffer agents and cryoprotect agents. So this annotation helps you to filter through the ligands that you need to concentrate on. And I think this is the most interesting part for me, is that like once you know the ligands as a structure, from structure, which structure you need to look for, that's what we consolidate those informations. We have a table which shows all the proteins the ligand binds to, and all the corresponding structures, how many structures we have, and corresponding annotations in that structure, what is the likely function of that ligand. So, and if you are interested, once you see this information, you might be interested in a particular protein. For example, for hydroxylase, you want to know more about that protein. We have another page called aggregated views of protein. What this page does is we aggregate the structural information of a particular protein across the archive. So if you want to know which structures we have, how many structures in the PDB for this protein, what are the different type of ligands. And we also collect annotations from computation and experimental annotation from different resources. So that helps you to look at this information about that particular protein from the web interface itself. And this is another way you can understand about the binding of the protein. If you look at the 3D superposed view, it superposes all the structures of a particular protein and then cluster them. For example, in this case, we see 2 clusters, and from the superposition also you see that some conformational differences between different structures deposited in the PDB. Also, it also shows like all the ligands in the PDB, they are all binding in the same site. So that might be some active site you might want to look for. And in this case, let's see how can we look at, how can we understand more about the conformational difference. So it doesn't say like what caused the conformational difference, but at least it gives you a direction. Okay, there is a conformational difference, and should I be investigating more onto what are the questions I would like to, should I be interested in that? And in this case, I did check into the literature to see like the authors report that a substrate like molecule binding causes conformational changes. So this just to show that what that conformational change is like. So I did not download any structure here. I did not go to any web interface. Every image I've created is just using the web interface. So that's what I want to highlight here. And so if you are a little bit curious, like if I want to see how AlphaFold predicts, you don't need to leave to another page. Like we provide with a simple click, AlphaFold structure gets superposed and see how AlphaFold predicts this conformation. Which conformation does it predict? I was just a little curious which one AlphaFold predicts. So it just predicts some average of that. But PLDT was very low, so we can't really trust on that prediction. But it has much similarity towards the one without the substrate, which is most likely because we have more structures without that substrate in the PDB. Now, if you want to look at more on the binding interaction, you want to know a particular structure, how the ligand binds in that structure, you can come to the table again, and we have links to the PDB entry pages where we will show both the table-wise, where we show all the interaction of that ligand to the different residues. We also have the 3D and 2D views of those interaction. We also show the electron density, so that you are not tricked by some mistakes in the PDB. So if you know whether to trust that interaction, whether the ligand is, whether the crystallized model is in accordance with the electron density, you can see from the view. So in this case, we had closed and open conformations. So as you see in the open, like it's mostly interacting with the solvent molecules, but then we do have a lot of residues which are in the closed interacting with the ligand. Now, if you want to, that was interaction analysis within one structure. But if you want to know, if we aggregate the interaction information across the structures, so if you want to know which atoms are mostly interacting with the structure and to which amino acid is interacting and what type of interaction might be. So in the default view, we use a tool called PDB-RPGO. So to calculate, it calculates like 15 type of interactions, and the default view shows the aggregated information of the entire interaction type. But then you can also filter the type of interaction. If you are looking at like particular type of interaction, those statics will get updated according to that. And these are all interactive, so you can, if you are following along, you can check like how does it vary. I don't know if this one ticks. Yeah, so this one is just showing like if I update to aromatic interaction, everything gets updated, all the values get updated just to include that type of interactions. Now, if you want to look at what are the similar ligands present, so once you know about a particular ligand, maybe you're interested in like what are the similar ligands already present in the PDB database and how similar are they, what are the different type of substructures present, we highlight them in the interface. And we put a cutoff of like a 60% similarity minimum, and then we also highlight which part of the ligand that we have the high similarity. And we also show stereoisomers, and we also give the links to the entries from which it came, so that if you are interested in that particular entry, you can navigate to those web pages and then go through different information provided in the web page. And so we can't provide all the information related to the ligand. So there are dedicated resources for small molecules. So we provide mapping to different databases. So you can navigate, for example, if you want to know bioactivity data, you can navigate to ChemPel or PubChem, or like if you want to know more about the drug-related information, you can go to Drug Bank. So we provide linkages for all those databases within the web interface itself. Now, some of the works we done that's still not available in the web interface is the integration of the bioactivity data with ChemPel. So this is one work we done, the data science from ChemPel Melissa. So what we did is we integrated the bioactivity data. We have the bioactivity data in the ChemPel. So ChemPel targets, they map to UniProt ID, and we also have PDB structures mapping to UniProt ID. So we have one link between them. For the small molecules, we can represent this small molecule using the same identifier like INCHI, and then map them using these two mapping. We did the mapping between PDB and ChemPel, and we got almost like 17,000 PDB protein ligand complexes corresponding to 39,000 bioactivity data. So we provide this data for you to download from our FTP area. We are currently working on how can we best showcase this information so that it's accessible within the interface itself. So this is just to show in this particular use case I have been discussing about H4B, you can use this information to identify and do analysis. This is, we created some training materials on that. So if you go to the PDB GitHub page, you could get this training material available to give an idea like how can you use this data for some kind of preliminary analysis. And another work we did with CCDC is an integration of small molecule data with CSD. So this was the project I had worked on last year. So we integrated the small molecule data. So each resources concentrate on different aspects of CSD. Look at the crystallography of small molecule data. We can look at the bioactivity data. And in each website, they represent the data differently. But for small molecules, you can at least represent them in a unique way and link them. So we use the source called Unichem, which is maintained by Campbell, to do the mapping between these chemical resources. So now we have almost 3,000 molecules mapping. So we went with a strict mapping of like complete mapping of InchKey. As you know, sometimes there might be salt molecules or maybe stereoisomer differences, but we have not taken for that. So if you relax that, you might get more molecules in this. Now, one of the work we also did was integrating the fragment hotspot data. So this is one work done in CCDC. What they have developed a pipeline to identify areas in which we might see most likely ligand binds. So idea is to you take a structure and then run a pocket identification algorithm. So in this case, they use the software called GeekCom to identify the pockets, and then use different type of fragments. In this case, three type fragments with properties, a polar acceptor and donor, and then you place these molecules inside that, then do different iteration of rotations to see which sites at which these fragments are binding, and then create a map of the areas where the fragments is likely to bind. So we are currently in the process of how can we create this data in a weekly process, and then how can we also showcase this data and also map to already whatever we have available in the PDB database. We have structures with fragments, whether there is a mapping between this calculator and the fragment structures we have available. So we are working on that. So this is just one part of ligand information I have shown. We have different pages for the proteins, complexes, and we are still working on how to improve these resources so that you can make use of the tools we have available. So for you to just, all the data, so when we create a web interface, it has like, if I want to say like it has 3-4 process. So you have the data, then you have the tools which process the data, then you need an infrastructure to connect this data, and then you have an interface. So the web interface I explained, but then if you want to do batch analysis, you have APIs available. So for all the data fetching is done through API. So all the data fetched in the website, you can also fetch this data using the APIs. So if you are interested in a multiple ligands, you can use these APIs to call and fetch the data. So for the ligand analysis, we use the one tool I mentioned, PDB CCD utils and PDB relic. This is also an open source package. So this is what we use to do the annotation, like cofactor and drug-like annotations based on similarity. PDB Arpeggio. So Arpeggio is a software developed in Tom Blundell's team in Cambridge. So we took that software, and currently we are maintaining that software. So we updated to pip installable Python package. It calculates different type of interaction between, and we do run this for all the new entries come into the PDB in a weekly process. And the PDB bioactivity, so this is soon to be released as an open source software from Campbell. So this one maps between PDB and Campbell database. And Unichem is a source which you can use to map between different resources, mainly maintained in Campbell, but there are other resources like PubChem also within the Unichem. And Hotspot, this is an open source software that I mentioned about, you can use to find the fragment hotspots in the structure. Now, we use different type of databases. So one is the graph database, which we also, so if you want to get the entire data, whatever we release, we have a dump of GraphDB, but it's huge. But we also have the Oracle database, which mainly cater to create the APIs. And the web interface I showed, like you can now, if you want to do some preliminary analysis, you can use this web interface to navigate the data. So we are currently still improving on that. For example, PRD and CLC, we don't have all the features I have explained. So we are improving on that. And we are working on how to visualize the different type of data. So we dedicate a lot of time in thinking about how to best visualize this data, how can this answer different questions. And that's where like we would like to get inputs because like based on different people's background, like the way people look at the data and the use cases varies. And we would like to know what you want to do. And also, we are also looking at like right now, the search capability is limited, but we are working on how to improve the search. Then that's also an area like we would like to know how do you want to search, like what are the different use cases you have seen, what are the different pain point you have seen when you wanted to analyze the data from PDB. So if you are kind enough to scan this QR code and give us the feedback, that would be really lovely. And we make our development user driven, so that if you get the feedback, that could be discussed and then we could cater to your needs. So thank you so much for listening. And none of this would have without possible. We have a dedicated team of curators, developers, and like UI UX experts, postdoc. So it's an effort of a team. And of course, without the funding, we can't do things we love to do. So thanks for the funding and thanks for listening. So if you have any doubt.
Any questions? So
this is really a question about deposition. Do you know if you can update your deposition once you've done it? Because normally you publish a paper and you say you stop refining when you publish the paper. Then you spot some error a couple of years later or something, you want to redeposit. You don't want to change the reference in the original paper. So do you have versions? Versioning would be good for PDB.
Yeah, okay. I don't work on the deposition part, and that's why I have my colleague Deborah here. So she could answer that.
Is Deborah here? Yeah. Do you want to answer it right now?
Yeah, we do have versioning. You can request and log request, re-upload your model, and that's it.
All right, thank you so much.
Sorry, we have one more.
Yes, Gerard Bricogne, global phasing. Sorry, about the last slide here. So it's a trivial remark anyway, sorry. There's a little diagram just left of Google DeepMind on the second line. Doesn't it have its helix going the wrong way?
Okay. I will take that good feedback and let them know. I don't know which logo it belongs, but yeah. Friday afternoon, still people are awake. That's good.
Okay, so our next speaker is Gustavo Lima from Aztec Pharmaceuticals. So,
okay, so the final speaker for today and for this meeting is Melanie Oelke from Helmholtz Zentrum Berlin, also known as BESI. She's responsible for the fragment screening facility there, FTEX. And I will leave a little pamphlet here if anybody wants to pick that up.